There are two ways to classify a bacteria, which is by putting them into the categories gram-negative and gram-positive.
The only way to decipher what the gram status of a bacteria is to run a gram stain. The first thing you do is to get bacteria and put it on a glass plate. And then after that, you have to fixate it, which means you kill all the cells, and you do this by using a Bunsen burner. Next, you have to use crystal violet, which dyes the cells purple. Then you have to do an iodine treatment. This will make it darker and stick the crystal violet to the peptidoglycan layer. Next is decolorization. Any gram-negative bacteria will lose the purple color from this step. Lastly is the safranin process, which will dye the membrane in gram-negative bacteria red or pink and gram-positive purple.
Gram-positive bacteria only have one cell membrane and a thick peptidoglycan layer on the outside. It also has teichoic acid, and the role of teichoic acid is unknown, but it is very important to the bacteria. Gram-negative bacteria has two membranes. Gram-negative bacteria has two membranes and a thin peptidoglycan layer between the two membranes. Gram-negative doesn't contain teichoic acid, but it does have LPS. Gram-negative bacteria is more susceptible to lysis.
Once you know a bacteria's gram status, you can also classify it by the morphology, or the shape of the cell. There are three main shapes, and that is cocci, or spheres, bacillus, or rods, or spirals.
Cocci means a single sphere-shaped bacteria. Streptococci means a chain of bacteria. And staphylococci means a clump of bacteria. Bacillus is a single rod-shaped bacteria. Streptobacillus is a chain of bacteria.
You may be asking yourself, why do I need to know the gram status? Well, it actually is very important. So, when you are prescribed an antibiotic, some antibiotics only target gram-positive and some only target gram-negative. So that is one example of why they are very important. But to further continue to learn about bacteria, you can run a set of metabolic tests, which will help narrow down what your unknown bacteria is. But the first step is to always run a gram status test, and that will quickly eliminate your options once you know if it is gram-positive or gram-negative, since they are so structurally different.